Okay, Joseph, go ahead.
Hi everybody, my name is Joe Barrett. I'm a junior at Mountain Vista Governor's School, and my project tested the effects of vibration on microbial growth for the purpose of increasing the degradation rate of polyethylene. And I'm not actually the first person to see whether increasing microbial growth can increase the degradation rate of plastic. In fact, there are 3 leading scientists across Australia, New Zealand, and and other parts of the world who have used chemicals, changes in states, and simply different types of microorganisms to increase counts of microbial growth. However, the toxic waste has been too great of a side effect for their research to continue. Most notably, however, a young Canadian scientist named Daniel Berg was able to increase the degradation rate to only 3 months, which is outstanding. And he did this through using his own re-inoculation technique and through isolating microorganisms. Daniel Berg became my inspiration for this project, as he was a high schooler when his technique was discovered. And as I read through his research, I wondered how vibrations could impact his work. So you all are probably wondering, why does any of this really matter? Well, being able to increase the degradation rate of plastic is huge in the overall scope of pollution. It's estimated that landfills contain upwards of 6 billion tons of pollution, or 6 billion tons of plastic for up to 10,000 years. So virtually every single piece of plastic that's been made still exists in some way, shape, or form. It's also estimated that in the oceans, there are 5.25 trillion pieces of plastic debris floating around harming marine wildlife. And if that's not bad enough, it gets worse every year, with more than 35 billion plastic water bottles being used just per day, and 500 billion plastic bags being used just per day in America. So reducing the plastic footprint is obviously huge in the overall scope of pollution. So the purpose of my project became increasing microbial growth through the use of vibrations for the purpose of increasing the degradation rate of plastic. And this year's research only focused on the first part of that, which is using vibrations to increase microbial growth. So my question became, can vibrations increase microbial growth? And my null hypothesis basically stated that when vibrations are applied, there will not be a difference in microbial growth, while the alternative hypothesis said that there will be an increase in microbial growth. And the materials that I used were pretty basic. I used landfill dirt from a local landfill. I used Bushnell hoss broth, which is a mixture of chemicals, as a growth medium in every single one of my trials. And I used polyethylene as my source of plastic. And I got that from a local grocery store as the grocery store bags. And then I used various glassware, an autoclave spectrophotometer, and cuvettes from Fauquier High School's biology lab. So once it came time to start my project, I had to identify my variables. My independent variable became basically what my experimental group was, which was vibrations provided through the orbital shaker at 150 rotations per minute. And my dependent variable was the amount of microbes produced, which was a representation of the absorbance. And I didn't take a single measurement until 48 hours after the start of my experiment, and Bushnell hoss broth was used in every single trial, and the lab setting never changed. And as per the amount of trials, I had 6 total test groups with 30 trials per each to allow for statistical testing. So once I had all my materials, I began my methods. And the first part of my methods was to actually determine my test groups. So I wanted a control group and an experimental group. And my experimental group had vibrations applied at 150 rotations per minute, while the control group was simply left alone, so it just sat. But they both had the same 3 test groups, which ended up being Bushnell hoss broth, or BHB, as you see up there, and BHB, or Bushnell hoss broth with landfill dirt, and then Bushnell hoss broth with polyethylene. And once I had this figured out, I measured my materials in the test tubes. and then 48 hours later, I measured the absorbance using my spectrophotometer, and the greater the absorbance, the greater the microbial growth. I measured the absorbance at 3 different wavelengths based on the preliminary experiment and literature findings, and this gave me 540 data points to analyze, and I analyzed it using a one-way ANOVA test and a follow-up post-hoc Tukey test. So these are the 270 results for no vibrations, and these are the 270 results with vibrations. When it actually came to analyzing the results, I restructured the raw data into categories based on wavelengths to help eliminate any lurking variables. And then, so that led to 3 ANOVA tests, which all returned very, very significant results. And then for my post-hoc Tukey test, which actually compares each test group to each test group to each test group and so on to determine where that significance occurred, it said that at the wavelength of 403 nanometers, the Bushnell-Haas broth with landfill dirt with vibrations produced microbial growth at a greater rate than without vibrations. This is just a graphical representation of the Tukey test. So significance is denoted by an asterisk, and as you can see, significance only occurs where the standard errors do not overlap. These are the same 2 graphs from before put side by side. So as you can tell, there is a more general, or generally a more positive trend in absorbance or microbial growth when vibrations were applied as opposed to when no vibrations were applied. And that was basically my analysis of the raw data. And I was able to say that vibrations increase microbial growth more efficiently than with no vibrations, and I was able to reject my null hypothesis. So there is sufficient evidence to say that vibrations increase microbial growth. All 3 test groups did have significant, or not significant, but they had increases in microbial growth. However, the most effective increase was in the test group of Bushnell-Haas broth with landfill dirt with vibrations. In conclusion, my null hypothesis was rejected, and microbial growth was amplified through the addition of vibrations. And to answer my research question, vibrations can increase the rate at which microbial growth occurs. So after all of my research was sort of over, I asked myself, why did this happen? And I came up with this. So basically, when bacteria grows, there's a lag phase, a growth phase, or exponential phase, and then a stationary phase. So the reason that I chose to measure 48 hours is because, based on the literature I read, the log phase or exponential phase usually lasts for 48 hours. So I chose to measure at that peak right there. And my findings basically shortened this lag phase and increased this exponential phase. So whether you're shaking a glass of wine or increasing a chemical reaction, swirling ensures a good way to aerate any type of mixture, and the orbital shaker was a good control as it was a mechanical process. Also, oxygen has limited solubility in liquids, so the liquid was allowed to be aerated through and through as it kept swirling and swirling. So, and for further research, I would like to test the results of this research, which would be vibrations, with the 3 leading degradation techniques, which are again chemicals, changes in states, and simply microorganisms. And a real-life application that I see this project being applied to is a landfill, because in landfills today, there's little to no movement of dirt whatsoever. So if we could redesign landfills to have underground vibrating rods that increase the microbial growth, then that could increase the degradation rate of the plastic in the landfill. And I'd like to say thank you to my mentor, Dr. Swanwick, for all of her knowledge and lab space, and also to Mrs. Ribeiro for her countless meetings and discussions about this project. And I could not have done it without the Fauquier County landfill dirt. And also to my mother for her camera assistance. So, any questions?
I got a very good talk, and I really wanted to go back to your data and see that 2-key analysis.
Okay.
Because I saw something interesting.
Mm-hmm.
Can you flip that too?
Okay.
There you go. So when you did the differential analysis between your groups, plastic was not significantly different whether you vibrated or not.
Right.
And I have a little trouble understanding, you said you can use it in the vibration technology in landfills.
So for future,
So how does that?
for future research, I'd really like to test using landfill dirt with Bushnell-Hoff's froth, with vibrations, with plastic.
Mhm.
So basically combine these two
Mhm.
to make almost a fourth test group. Because the way that my growth medium worked was the introduction of carbon. So my theory onto why dirt had a greater bacterial growth is because there's a lot more carbon in landfill dirt than just
Mhm.
plastic alone.
Mhm.
So, any other questions?
So it was interesting that you anticipated. I was going to ask why. You said you might want to know why I think this happens. So you talked about aeration. I wonder if some of the bacteria though buried in the landfill are anaerobic bacteria.
I'm not positive, but this was taken straight from a landfill, and my test tubes were left uncovered. But for future research again, next year possibly, I'd like to possibly cap test tubes to then measure the gas output or something, just
Probably
then measure the microbial growth that way to see if I come up with different results.
Mhm.
So like you said, if it's not, or if it's anaerobic, I guess.
Would you consider where do you sample in the landfill? Like from where in the landfill you will collect your samples? Is it
I mean,
make
once
a
you bring
difference
it
once you
above
bring-
ground, right?
yeah.
You probably select for the aerobic ones, and the
Yeah.
anaerobic ones aren't where they're gone. So
If
your
you-
study is on the aerobic ones at that point because they're not there if they were the anaerobic ones, even if they were there in the landfill.
Right.
But I just thought when you buried something to create vibration, that I'm thinking layers down, are you really dealing with aerobic ones? And then
Okay.
vibration make a difference, but.
That's a good question. I'll have to research that more.
It's cool.
Thank you very much.
Thank you,
right,
everybody.
everyone,
That was
thank you.
really good presentations, and everybody did an awesome job. We
Right.
really appreciate you all coming up here and sharing your knowledge. Very well done. Thank you very
We
much.
appreciate the audience staying around.
Yes,
really
and asking
engaged.
questions
That's
and-
great.